while we are waiting for Judith Sheff, let me give a very brief introduction of the speakers. Dr. Robert Laumbach, MD, is Associate Professor of Environmental and Occupational Health and Justice, Director of the Community Outreach and Engagement Center for Environmental Exposure and Disease at the Rutgers School of Public Health. Dr. Peter Jaffe is William L. Knapp, Professor of Civil Engineering and in the Department of Civil and Environmental Engineering at Princeton University. Dr. Arjun Venkatesan is Associate Professor of Civil and Environmental Engineering and Director of the Emerging Contaminants Research Laboratory at NJIT. Dr. Dip Sarkar is Professor of Environmental Engineering and a founding director of the Stevens Center for Sustainability at the Stevens Institute of Technology. And Michael VTS is right walking in there, Vice President for Entrepreneurship, New Jersey Innovation Institute. And our fearless leader, moderator, Judith Sheff is somewhere around here. But as she walks in, let me request to start. And our first round was for you to give a brief presentation about your vision, your thoughts, and what your recent accomplishment, because the last session was a good segue of saying, let's say, and one of the questions that was asked by the audience, that how do you create a translation of the technology R&D to benefit the society? Not in a specific order, but maybe I would request Peter, actually, hold on that particular thought, because I'm just thinking about the need. So I think it would be good for Dr. Laumbach to get us started about what his findings are from the public health point of view. Doctor.
Well, as a couple of speakers have already viewed, certainly there are many health effects, many adverse health effects.
Do you have a presentation?
And we have, yes.
Yeah. Could you put presentation? Yeah, there it is. Okay.
Well, you have to turn it on, I guess.
Okay, very good.
All right.
The health scientist viewpoint on what I think our panel, how it sort of comes together conceptually, in that go from detection to degradation and removal of PFAS, and then scaling up those processes and then commercialization. And then health, and what we're doing that for is to have a health impact, right? And I'm going to talk a little bit about getting the health in a little bit earlier, like maybe at this point at which we're going to be doing intervention, when we're scaling up and commercializing what might be called a natural experiment, which we're going to be reducing exposures to PFAS, and we, as health scientists, can utilize that to study health effects. And I'm going to talk about a particular case study, Paulsboro, New Jersey, that allowed us to do 3 different types of studies, and I'll review those, but looking at health effects. Paulsboro, starting in 2009, learned that they had a problem with PFAS. It was going on decades before that. But in 2009, PFNA, the 9-carbon legacy PFAS, was found in the drinking water at 96 parts per billion. And then in September 2013, more sampling of the drinking water found levels of over 150 parts per trillion. which was the highest ever recorded. And then there was an advisory in 2014 that infants shouldn't be drinking the water. And then shortly after, in April of 2014, the contaminated well, which was the only well supplying the town at that time, was shut down. Another well was brought back online. And then in June of 2016, granular activated carbon was installed on the contaminated well. So the first study that we did was actually taking advantage of a lawsuit which led to 1,300 participants in Paulsboro having their blood tested for PFAS. That's all they got was a blood test. No interpretation. They got the lab report. So an investigator at Rutgers, Judith Graber, and a team of us did a study in which we invited 194 residents to give us their results. And what we found was that for the 4 legacy compounds that were tested, that PFOA and PFOS were similar to background. So this bar chart shows compared to NHANES, which is the national sample at that time. And then PFOA, a little somewhat elevated, but PFNA was 4 times the national average. And it would have been higher probably, except already people weren't drinking the water in Paulsboro for some period of time due to some prior issues they had with VOCs in the water and some other contamination issues. So the second study that we did was to look at the effect of the shutting the well and treating the water on blood levels of PFNA, and with 120 participants who had 3 samples over 2 years. This bar chart shows the decline in blood levels of PFNA in those same subjects, repeated measurements, indicating about a 4-year half-life. There hadn't been a lot of studies done yet about PFNA and its half-life. This was a valuable contribution. And then I'm going to talk about a health study that we did in Paulsboro, where we had participants that entered the study in 2021 to 2023 and found the levels even lower, as you can see in the box on the right. So it's already been talked about with the various health effects of PFAS, and they start in utero, in child development, and adulthood. So I won't go into too much detail. Just to mention a couple of things that we're doing in the present study, like one is cholesterol. So a lot of studies have shown an increase in cholesterol, but there is a sort of a naysayer or a skeptical argument that cholesterol and PFAS may go together in that they're like recirculated from the gut with bile. So when we do cross-sectional studies, like the one I'm going to describe, we're measuring PFAS in blood and cholesterol at the same time. So we don't know really which came first necessarily, or if they're just sort of going along together for the ride. So I want to talk a little bit about sort of how we can sort of try to answer that question. But first, our study is one of 8 across the country. It's a multi-site study in which 6,000 adults and 700 children were recruited all across the country. A very in-depth sort of a study in which we, in Paulsboro, had 776 participants and 88 children. And they had to be residents in Paulsboro between the years 2005 and 2014. We measured PFAS in their blood, 7 different PFAS, did interviews, questionnaires about their health, blood tests that include not just cholesterol and blood glucose, but other, essentially over 100 different biomarkers related to health. And then neurobehavioral studies in children and adolescents, and then medical record abstraction, when we could get medical records, which was not easy to do. And then now we're also reconstructing past exposure at each of the sites based on history of water contamination, pharmacokinetic modeling. And then we're doing studies right now. And initially, cross-sectional studies, which again are measuring health effects at the same time that we measure PFAS. So the studies we have in progress include looking at, again, cholesterol, blood lipids, thyroid function, thyroid disease, diabetes and blood glucose, blood pressure, metabolic syndrome, heart disease, obesity. Again, it's amazing really how many different health effects there are related to PFAS. An inert molecule, right? I mean, biologically, essentially inert, these long-chain PFAS. So even that's still being, sort of worked out. The only publication we have so far is this methods paper describing what we did. But I wanted to mention one thing that could help with this problem with cross-sectional studies of like the chicken and the egg, which came first, is that if we have opportunities through interventions, whether if there's a large project that's going to be reducing levels of people's exposure to PFAS, that if we get a sample of blood and some basic information from people, that's not, of course, not easier said than done. We could then have information at the time when people are exposed and then look for effects of the intervention. So reducing exposure in a large number of people and then seeing how that influences people's health. Because we really can't do the opposite, right? We can't expose people intentionally. That would be the gold standard for detecting whether or not PFAS is actually causing a health outcome. Obviously, we can't do that. But we can take away the exposure and have also better information related to causation. So there's a couple other things certainly that we need in terms of looking at health effects, the effects of mixtures of PFAS. They're always occurring in mixtures. We need longitudinal studies. Again, more studies looking over time. We need better like high throughput methods. We can't rely on animal studies, both because they're very inefficient, as well as the fact that the animals are very different than people when it comes to PFAS in terms of half-life and other, especially pharmacokinetic, we call them, differences. So that's what I had. So thank you very much. We get the slides.
All right. What I want to do is give a quick overview how in academia and science you can go from a by chance discovery to follow it up by hard work to understand the process more, and then as engineers to eventually develop a technology. It's a challenge. I'm going to try to do it in 5 minutes, 20 years of work, a big team, but let me race through. So 20-some years ago, we did some work in the Pine Barrens, and we discovered this process that has been later called PMOX, where ammonium is oxidized under iron-reducing conditions to nitrite. We then went out and we discovered the organism that's responsible for this is an acid-bacterium, it's called strain A6. What is interesting when we look more at the sequence of this acid-bacterium, it had genes for a series of reductive dehalogenases. It also had a gene for fluoride pulp. So we then went and looked at, is it able to degrade chlorinated and fluorinated compounds? And we have done these kind of incubations in the lab, very large sets of incubations. As it uses iron as an electron acceptor, so we see these orange iron cloudy colors. We do those incubations. We did them with PFOA, all kinds of linear alkyl substances. We see that we can degrade PFOA, hepta, hexa, penta very slowly, and perfluorobutanoic acid. Same thing for the sulfonates. And on the right side, we see that we're producing fluoride while we're degrading these compounds. The bottleneck is in the 5-carbon, the perfluoropentanoic acid. It does degrade a little bit. We see production of fluoride, but there's some steric hindrance that the organism really has a hard time degrading it. When we look at PFOA, for example, as I degrade PFOA over 2-month period, we see the production of HPFOA, that is PFOA with one hydrogen, with one fluorine substituted for hydrogen. So that's the first dehalogenation that happens, and then other organisms can break it down further. And then we have a decarboxylation, and we see the production of the smaller perfluoroalkyl substances. Same results we see for the sulfur compounds. So this is the background, and now we're looking at different technologies that we're developing. Again, when we look at these incubations, what we see is that we try to track amount of fluoride produced with the expression of the different reductive dehalogenases. There is on the left side fairly novel reductive dehalogenase that has a very strong linear correlationship between the amount of fluoride we produce and the amount of expression of this reductive dehalogenase. Other ones are very scattershot, and this is probably a co-expression. I'll get back to this reductive dehalogenase, which I call RDHA, in a little while. So one of the challenges that we had to overcome first is that this organism grows in mud. We need a slurry with iron. It's very hard to run a bioreactor for a long time with a lot of solid iron phase. So the first phase that we looked at bioelectrochemical reactors in microbial electrolysis cells. The organism is electrogenic. It can transfer the electron to an anode. That anode then with a slight potential transfers the electron to a cathode where you have hydrogen formation. And we have seen when we run these microbial electrolysis cells on the left, have for pure culture and enrichment culture the amount of current that we produced, and the very on the right side here is the amount of PFOA that we're removing. So in I think it was 18 days or so, we remove about 60% of PFOA in these microbial electrolysis cells. The rest are controls where we don't remove any PFOA. So that's one technology that we're working on. Like batteries, microbial electrolysis cells are not easy to scale, so it's going to be a challenge to come up with a reactor that can treat a few thousands of gallons per day, but it may be doable. The next one is collaborating with our group in material science, and they developed hydrogels that we then impregnated with iron and ammonium, which the organism needs. One of the hydrogels they produced, actually, when we put it in contact with the bacterium could get into the hydrogels. Here I have pictures, and we have some little samples here in the back of our posters. And then we did so far only batch incubations with these hydrogels, but here I see the amount of PFOA that is being removed on the left in the hydrogel with the organism, and the right is the hydrogel without the organism. There's no removal. And the red bar is the amount of fluoride produced. So we are able to produce these hydrogels that are impregnated with ammonium and iron and the organism, and use them as an immobilized phase to degrade PFAS. So this is sort of the bioreactors. Next, we want to go at bioremediation of field sites. So here I'm doing our muddy incubations, and that's why I say we need a different kind of bioreactors. So on the left, we have sediment from AFFF impacted sites. We know how to stimulate the organism when it's present. And the right, we do the same thing in column experiments. And what we've seen, for example, is that as we stimulate the organism in these columns, we can degrade PFOS quite effectively. So slide D, Series D, is where we add all the nutrients that the organism needs to be stimulated. We add iron, and we see significant amount of PFOS degradation. Slide A is where we don't add anything, we just DI water to the mud, and the organism doesn't become very active. So we are at a point, and we've been discussing also with C.M. Smith, Charles Schaefer, who is here, and we're ready to go to a field demonstration because we feel we understand this process good enough that where we know the geochemistry at an AFFF impacted site, where these organisms can be present. They may be dormant, but we believe we know how to stimulate them, and then we can sort of degrade these PFAS. The question is we don't quite know yet how low we can get over time. We are pretty certain that we can reduce 90% of the mass or so, but to what levels, what the final endpoint is, we're not quite sure. Finally, and extremely exciting, is I showed earlier that there's a strong correlation between the reductive dehalogenase and the amount of fluoride produced. So I got in contact with a colleague that's here, Professor Avalos, and he took that gene and inserted it into fast-growing host. They can grow it overnight. produced the enzyme, and then we applied that enzyme directly to a series of matrices with PFOA. The next slide shows on the left the left is soil from AFFF contaminated sites. So we produce the enzyme, we apply it to AFFF contaminated soil. We have 20, I can read from here, 25% removal of PFOS in 24 hours. We apply a second little dose, and it goes down to almost 50%. On the right side, there's a series of activated carbons that we loaded with PFOA and other matrices. And we can also add that enzyme to activated carbon. And we see that we can degrade the PFOA that is sorbed to the activated carbon, hopefully coming up with a method of regenerating activated carbon. And then the middle panel is sort of intriguing. We had several of the fluorine-based polymers where we added that enzyme, and we see that as we add the enzyme to the polymer, we see the production of fluoride. The amount of fluoride produced is not large, but it seems to even attack at least some of the fluorinated moieties in these polymers. So Princeton has a series of patents or patent pendings in these processes that I described, so we're ready to work with any of you that is interested in any of these processes. These are the folks that are all here today, and we have e-posters. If you see anyone have questions, please feel free to ask. And I'll see you then. I have the glass.
I guess it's me next. Well, slides. So while we wait, I know we're already running late, and today I'm going to present more about an overview of PFAS and maybe add some new information from what we already heard since morning, and we had some really great talks and great presentations. I'm Arjun Venkatesan. I'm an associate professor here at NJIT. I always like to start with this slide about PFAS because we did it and we thought it's really pretty, but it also says like PFAS is pretty much in all streams, everywhere, any matrices that you detect, and we kind of say that PFAS has become a new element in the water cycle, in Earth's water cycle. So that's a huge statement when it comes. And it's not just a statement because more and more research is proving that it is true. I don't want to spend too much time on what we do, but I just want to highlight some of the things that we're doing with regards to PFAS. So we do quite a diverse work in Marlborough at Emerging Contaminants Research Lab. Many of our students are here, we're doing the hard work. So we assess what happens after an AFFF spill, like how quickly it spreads into the environment. That's something that we do. We also look at how PFAS accumulate in organisms like crabs. And we also recently published a study on shark tissues. And sharks also have PFAS in them, like especially the ones that swimming nearby the coastal regions. We also track PFAS, how it moves through wastewater treatment facilities. We have studies to look at biosolids that's applied on land, and that could be reintroducing PFAS into the environment. And in addition, we're also developing technologies to treat PFAS and also to destroy PFAS. So essentially, I wake up thinking about PFAS and go to bed thinking about PFAS and also wonder how much PFAS I get exposed to. But I want to highlight something, why it is so difficult to treat PFAS, because we've heard so many times that PFAS is not just one or two chemicals, it's a family of chemicals, like more than 15,000 different formulations. And unfortunately, each of these PFAS behave very differently, right? So if you design a technology or a detection process, picking one PFAS, that's not going to be applicable to other PFAS. So this makes it very tricky, right? So you have like ultra short chains, you have partially fluorinated PFAS, you have long chains, You have precursors that we don't even know the structures about. So it's getting really complicated, and we have to think holistically how we're going to address this issue. Yeah, as I mentioned. And what we detect PFAS is just only a handful of contaminants, right? So the EPA method has like 40 different PFAS that we can detect. We have additional standards to look into to look at more PFAS. But this chart kind of tells you the total fluorine in the environment, and the PFAS that we're looking at is the yellow region, which is only a fraction of it. And we still don't have standardized methods to look at the larger fraction of PFAS. And one might ask, like, why do we need to care about these other PFAS? Because they're not regulated. But we have to, unfortunately, because some of these unknown PFAS in the bigger box can transform into the regulated PFAS. So if you just track the regulated PFAS with targeted methods, you may miss this out, and in your distribution system or during your water treatment process flows, these precursors can be transformed into detectable PFAS, and when you open the tap, they may have levels higher than what it's being regulated. So it is important to know the total extent of PFAS in taps. Oh, I didn't even realize the slides moved on. Okay. So something is off with my flight. It was a very nice figure that I generated. It's kind of missing. But anyway, the point of this slide was like, it's also like very difficult to destroy PFAS. We've heard time and again that carbon-fluorine bond is one of the strongest bonds in the universe. So many of the destruction technologies that we have developed only look at targeted PFAS. So you see like for example, PFOA in the before treatment, and after treatment you have like no PFOA, and that's not a success, unfortunately, right? Because PFOA can transform into other PFAS. Maybe we're not detecting. So you need to close the mass balance there. So you're maybe producing partially defluorinated PFAS that we can't even detect, and they may be still toxic in the environment. So you need to focus on closing the mass balance on destruction approaches. I don't know why my slides are behaving very weirdly, but so this is a summary slide where I kind of try to capture my thoughts about what are the needs in the detection, capture, and destruction of PFAS. And I think it's a challenge in all aspects of it, because detection, PFAS occur at very, very low levels, right? Right now, the gold standard to measure is using mass spectrometry, because that gives you a precise and sensitive technology, but it also takes time to measure PFAS. So like many of the municipalities complain about not getting the data quickly. It takes like 2 weeks to get. So like many of our colleagues here are developing detection technology and sensor technology to go up to the field. But again, it's all working great in the lab, and we are figuring out a way to translate this research into field applications. And that's immediately. Regarding capture, activated carbon, ion exchange resins, they all work great, but they have their own limitations about whether they capture all PFAS. Do we know enough information about capturing these unknown PFAS, because they may be transforming into detectable PFAS? Those are some questions. And of course, there are many adsorbents available. We also need to think about the cost. In New Jersey, it just shocked me when I got my first water bill, because I moved from Long Island, I was paying $30, and here I get a bill for $250. I'm like, wow. And we don't want to burden the customers with additional fees, so we need to think about more cost-effective and practical solution for PFAS. And with destruction, again, time and again, we need to focus on completely closing the mass balance, not just like disappearance of like detectable PFAS, but you need to monitor the organic fluorine to make sure that it's completely destroyed to fluoride and innocuous chemicals, right? Again, I don't know. So I had this really nice activated carbon filtration, but it went missing. So I want to highlight like 2 technologies that we are currently developing, and I encourage you to stop by our station, and I can talk a little bit more. One of the technology that we are working on is improving the performance of granular activated carbon filtration for PFAS removal, because that's kind of the go-to technology. And we heard from Rich Calby this morning that they installed the first big system in New Jersey, but it also adds significant cost to them. So this technology that we are developing enhances the filtration performance over time and also saves a lot of cost to municipalities. That should translate into the users towards it. The other technology that we were focusing on also to have a destruction technology to chemically mediated destruction of PFAS. Right now, existing destruction technologies rely on really energy intensive and extreme condition processes like high temperature, high pH, and so on. We have developed a chemically mediated process that can destroy PFAS at ambient conditions. That's a big advancement, we feel, because it can be easily scalable. You can destroy PFAS on site, and this is more targeted on concentrated stream of PFAS, especially the residual AFFF, which the NJPFAS has taken so much of initiative in collecting and destroying them safely. We believe this could be a very cost-effective solution to do it as well. So that's all I had, and thanks for your time and